Today we are discussing the top non-COVID-19 AAC papers of 2020, a discussion with early-stage investigators. So what influential research outside of COVID-19 was published in AAC in 2020? Join me today and with my co-host, Dr. Maria Fernanda Mojica, to discuss the top 10 papers of 2020 with early-stage investigators. The objectives of today's podcast is to discuss pathways of young scientists in antimicrobial resistance, highlight important papers in AAC in 2020 that were not related to COVID-19, and stimulate discussion on important topics related to antimicrobial agents. Welcome to Editors in Conversations. This episode is brought to you by the American Antimicrobial Agents and Chemotherapy Journal, available at aac.asm.org. And I'm your host, Cesar Arias, Editor-in-Chief of Antimicrobial Agents and Chemotherapy. This podcast is supported by the American Society for Microbiology that also publishes AAC. Don't forget to check our latest issue of AAC, is the March issues, with important papers on resistant pharmacology of antimicrobial agents, epidemiology, and clinical therapeutics. The March issue features a history of AAC written by two previous editors-in-chief, Drs. George Eliopoulos and Dr. Lou Rice. I want to welcome my co-host today, Dr. Maria Fernanda Mojica, who is the host of the new ASM Journal Club focused on antimicrobial agents and resistance. Dr. Mojica is a postdoctoral scholar at Case Western Reserve University. Welcome back, Maria Fernanda.
Thank you, Dr. Arias, for your kind introduction and for the opportunity to share with you all this exciting news. So indeed, in 2019, the ASM established the Virtual Journal Club with the goal to increase academic engagement, community building, and collaborative networking. For 2 years, they have discussed papers regarding clinical and public health microbiology. However, this year, the antimicrobial agents and resistance and the virology community will be participating. As your host, I would like to invite you to our first journal club, Tuesday, March 16, in which we will discuss new approaches to combat multi-drug resistant bacteria. More information will be available at the ASM webpage.
Thank you very much, Maria Fernanda. And joining us to discuss these experiences, these papers, are two young stars in the field. Is Dr. Ayesha Khan, who is a postdoctoral fellow at the University of Texas Health Science Center in Houston, and Dr. Alina Iovleva, who is clinical instructor and Burroughs Wellcome Fund scholar at the University of Pittsburgh and the University of Pittsburgh Medical Center in Pittsburgh, Philadelphia. Welcome to the program.
Thank you.
I'm going to ask Ayesha first to give us a short introduction of how you got into research in antimicrobial resistance.
Okay, it's hard to make it short, but I think it started from where I come from. So I was born in Bangalore, India. So I pretty much grew up watching the real life impact of infectious diseases and understanding how it affected my community. So I wanted to do microbiology my whole life, pretty much. And I remained curious to understand how microbes worked, and ultimately also wanted to go into a profession that impacted, social change. So I came to America in 2011 for college at UCLA, and then started my PhD in 2015 and joined Dr. Cesar Arias' lab. And I think that's when I really fell in love with understanding mechanisms of antimicrobial resistance, because I saw that that was a direct way to translate my love for understanding the bug to the bedside. And ultimately, I found my passion for what I'm going to do for my career, which is pursue clinical microbiology through doing AMR research, as well as getting involved in patient care. So I think a combination of going on inpatient service, training in the clinical lab, as well as really understanding the real life implications that we can have by understanding mechanisms of antimicrobial resistance, epidemiology, and translating that in real time. We had the opportunity to work on some really challenging clinical cases. I think I really realized my life's purpose was to be at the intersection of both microbiology and clinical care, which is what I'm doing. I'm pursuing a fellowship in clinical microbiology that I'll be starting hopefully in a few months at Mayo Clinic.
Excellent. Alina, what was your pathway that brought you here?
So my pathways kind of started a bit later. I first got introduced to the field of infectious diseases as a medical student at Wayne State Medical School at Detroit, Michigan. Here I was first exposed to the specialty of infectious diseases, but also to patients with infections who had multi-drug resistant bacteria. And this initial exposure is really kind of inspired my career path through this day. I actually completed my internal medicine training at Case Western, where Maria's at right now. And I had an honor to work with Dr. Bonomo during my research elective as a resident. And basically, we had the patients with a really, really terrible resistant Pseudomonas. And I was at the time where ceftazidime tazobactam first got approved, and we were very excited to use it in the treatment of the patient. And I actually got to bring that bug to the lab, and we also found additional patients who were treated with this new agent. And I got to do a little case series investigating why this drug works in these clinical isolates. So that's kind of started, my love affair with multi-drug resistant bugs and figuring out how they work. So once I completed my internal medicine residency, I moved about 2.5 hours away to Pittsburgh to do my infectious disease fellowship. And here I've been, and I've stayed here for as a postdoctoral scholar and a clinical instructor. So I actually do about 90% time in the lab at this time. And again, I got to continue, here I work with Yohei Doi, and I got to continue to, bring the cases from the clinic to the lab to try to figure out the mechanisms and the best approaches to treating them. We have a lot of very complicated patients who get a lot of this resistant drugs. So I got to use, as a clinician, drugs that are still in the trial, the cefiderocol, meropenem-vaborbactam, which is very exciting. We also use some phage as well. So it's very nice to be able to kind of bring the problem from the clinic to the bench and to let it, science drive your decision making.
This is fantastic. Thank you very much for that pathway description. Now I'm going to hand it over to Dr. Mojica, who is going to lead us into the discussions of the top 10 2020 papers in AAC, non-COVID related.
Okay. First, thank you, Dr. Khan and Dr. Iovleva. Can you please pronounce?
Iovleva, but you can call me Alina. It's easy.
Okay. Okay. So Ayesha and Alina for joining us today. So for our reviewers, we selected the top 10 papers published on AAC in 2020 based on usage and citations. So Ayesha and Alina were given 5 papers each, and they will summarize for us the main message of the paper, the main results, and why is that important. So Ayesha, would you like to start, please?
Yeah, I think I'll start in order of time, just chronology based on when one of the earlier ones was, and then kind of go down the line. I think this is, so the first one we'll go through is titled A Biosynthetic Platform for Anti-Malarial Drug Discovery. This is by a group at the Imperial College in London and by Mark D. Wilkinson et al. And I think this was interesting to me personally because it's looking at a topic that we all read a lot about, which is novel drug discovery, the screen and understanding how it works. But using synthetic biology, which is the novelty of the paper, looking at using bacterial engineering. And in this case, it specifically uses bacterial engineering in E. coli to see if we can use that as a system to do complex drug discovery. And not just develop, but also test the efficacy of antimalarial drugs in a high throughput manner. And specifically, the issue that it's trying to combat is the rising rates of resistance in Plasmodium to all antimalarials recently, including the frontline, which is artemisinin combined therapies or ACTs. So what they do is they take E. coli and they engineer it to produce a novel compound, which is extremely expensive and difficult to produce. It's called violacine. Violacine, I think that's how you say it. But it's a violet indolo-carbazole pigment. It's produced in bacteria, and bacteria that produce it, example, Chromobacterium, they've been shown to be infectious themselves, highly infectious themselves, and that's partly why it's very difficult to make it, because you need advanced biosafety equipment, and all of this is very expensive. So they figured out a way to produce this in E. coli. And they go on to show in this paper that they can actually feed E. coli different tryptophan substrates to create analogs. So they end up coming up with, I think, like 68 different violacine analogs to test. And again, all of this is really fast and inexpensive because they just take the lysates. So they take the lysates and they're able to do in vitro killing assays to show, which is the notable result, I think, to ultimately show that these, all of these analogs of violacine have varying levels of antimalarial activity against both drug-sensitive and drug-resistant Plasmodium falciparum patient isolates, which hasn't been done before for a novel agent like this. And it targets both the sexual and the asexual stages, which I think is important in terms of resistance. And I think they go into also the mechanism of action, which is something that hasn't been looked at, I think, recently for antimalarials when it comes to novel drug discovery. And the mechanism of action specifically in this case seems to be inhibition of actin cytoskeleton, like it targets the actin cytoskeleton, but not polymerization, which means that it's specific enough to where it does have promising potential. And overall, I think it's important for anyone working on drug discovery because it shows that bacterial biosynthetic systems can be used to do high throughput screening. And it's much more cheaper and important for chemically intractable natural compounds, which I think are very expensive in general to produce.
Yeah, this is an interesting approach. As you know, there's been for a long time a lot of interest into harness genes that produce antibiotics and put it into work. But this is really a nice sort of harnessing of that concept to try to get compounds against something that is very important, which is more compounds against malaria parasite, which is developing resistant to that. Maria Fernanda, any other comments?
Especially because I have been working with two chemists, groups, they're trying to, we designed the drug and, with these complex ring systems, and it's very complex. And they found a way to, make E. coli do it for us, in a most effective way, cheaper way. And I think that's a pretty cool, system that should be implemented not only for anti-malaria drugs, but to other, antimicrobial agents.
Okay, let's move on.
Yes, Alina, do you want to continue with your other paper?
I will take over. So the first paper I will talk about is Tilarone, a broad-spectrum antiviral for emergent viruses by Sean Atkins and Peter Madrid. I found this molecule quite fascinating. So it's a small molecule, orally active interferon inducer compound. It induces alpha, beta, and gamma interferon. So basically, it helps your body fight the infection that you get. It's actually approved for human use in Russia and surrounding countries. As a broad-spectrum antiviral, so it's available over the counter for your upper respiratory infections, influenza, but it also has shown activity against hepatitis A, B, C, CMV viruses, other herpes viruses, and even is used as immunomodulatory medication for multiple sclerosis patients. However, it's been extensively studied in Russia, but never been evaluated and tested for safety and efficacy under studies that meet current FDA guidelines. So it's not really approved in US. So it's been shown to have activity in vitro against multiple different families of viruses, and most recently against Ebola. This paper adds another two potential viruses it can be used for. One is chikungunya, and another is MERS-CoV-2, which we all know is a cousin of COVID. And they, said they show a pretty good in vitro activity of this drug against these viruses, as well as limited cytotoxicity, making it a good compound for kind of for use. And they suggest that it's ripe for widespread testing against other emerging viruses, because we know something's going to come after COVID. We don't know when, but it's coming. So it would be nice to have something potent in our armamentarium. I think it's a good example of repurpose of an old drug for a new use. However, I wish there was more clinical data about, its safety and efficacy. It sounds like it's been around for quite time, and it might be very useful. And I think it's worth studying further. And maybe it appears that it's used against COVID as well, at least in some countries. So I would be curious to see if any kind of data on safety and efficacy will come out from that.
Yeah, great. I know we said non-COVID papers, but we decided to include this because I think moving forward, antivirals, specific antivirals or broad spectrum antivirals may play a very important role to keep people out of the hospital. I think one of the things that we learn in this epidemic is just keeping people out of the hospital, not ill, is a very important thing to avoid collapsing the healthcare system as happened during this pandemic. And I think we're going to see much more developing in drugs that you can do orally or for vials that is that are antivirals that are easily accessible and orally, that you can take it orally instead of IV, because that means you can take it at home and not go to the hospital. So I think this is an important example of how that could be achieved in a broad spectrum manner.
Right. And I think, that the COVID pandemic forced us to, look back at some drugs that were used in other countries and not here. I wonder, how long would it take for this molecule to, reach the public, light if we're not for COVID, for this emergency. So I think it's a good paper. I wish also that they have tested in other cellular, in other cells instead of just viral, because they say that this is a limitation because they don't produce, like interferon. So the activity must be much, potent, but it's not shown in here. But I like this paper. Okay. Ayesha, are you ready for your next paper?
Yeah, I'm actually going to switch it up and do a challenging case. I want to mix up the flavor that we have here. And also because the challenging clinical cases section is probably my favorite. So so this was a challenging clinical case that was an editor's choice titled Genomic Profiling of Evolving Daptomycin Resistance in a Patient with Recurrent Staphylococcus aureus sepsis. It is, I barely know how to say it because it is a new organism. And that's, I think, what makes this case pretty cool, because it is the first report of infection caused by Staphylococcus aureus. It is a novel staphylococcal species that is associated recently with invasive disease, but wasn't labeled, I believe, as a distinct species until 2015. So I found this really cool from both the perspective of understanding mechanisms of resistance, and because we work in our lab, in Dr. Arias's lab, on daptomycin resistance, and also from a clinical microbiology perspective. And I'm going to not give it away, but there's a 70-year-old woman who shows up, a history of end-stage renal disease, has prior toxic epidermal necrolysis from vanco, piptazo, cefazolin, and multiple arteriovenous fistula occlusions. So she went to the hospital, had a AV graft, shows up 6 months later with bacteremia caused by MRSA. And this is due to her tunneled dialysis catheter. So they do some source control, exchange that TDC, start her on 6 weeks of Dapto with rifampin, and comes back 6 months later with central line associated infection, bloodstream infection with E. faecalis this time. So 2 more weeks of Dapto, and then shows up a month later with MSSA bacteremia. And this is when it gets interesting. So this bug was identified with MALDI-TOF, which is pretty much the standard of care at most clinical microbiology labs to identify an organism, as again, methicillin susceptible Staph aureus. At this point, they actually removed the catheter, discharged with 4 weeks on dapto, so a lot of dapto, mostly due to the patient's prior severe allergic reactions. This is, I think, very unorthodox, I would say. And came back 3 months later, again, with MSSA bacteremia. And at this point, you just have this recurrent bacteremia with the same MSSA isolate being isolated. And the last occurrence is sepsis due to the MSSA, which is now dapto resistant, as well as vancomycin intermediate. And at this point, because of the odd resistance phenotype, they performed whole genome sequencing analysis and actually discovered that this was not Staphylococcus aureus, as the MALDI-TOF had called it, but a closely related relative, like I said, which is Staphylococcus argenteus. And it's pretty not that similar as I would expect. It's less than 90% identity, so it's about 89% identical. But because it's such a novel species, I don't think there's a lot of protein markers for the MALDI-TOF to pick up on. So I think it can be easily confused. And I think what personally the cool thing for me was part of whole genome sequencing is that you can pick up on hints for what the mechanism of resistance can be. And in this case, it was a mutation in MPRF, which is essentially this protein that can make a bunch of positively charged lipids and then flip them to the surface, repelling DAP from the surface. And it was a mutation that has been previously already identified in Staph aureus before. So I think this case shows a lot of interesting things. I think first, Staphylococcus argenteus being identified as a pathogen. Here in this case, I think that's something that people working in clinical microbiology labs are now more on the eye out for, especially because it was misidentified by the MALDI-TOF. And also showing the outcome of selective pressure from repeated courses of daptomycin in the absence of source control of an infected graft, which I think ultimately led to the emergence of both vanco and dapto resistance. So I think it shows the importance of source control. There's only so much that ID can do, which I know Dr. Arias talks about all the time. But I think it also shows the value of whole genome sequencing in personalized medicine, and if we have the capability potentially to be able to accelerate the process of, doing long and short read sequencing, that we might have been able to identify the species and understand that it's probably maybe evolving resistance to dapto. And I think what was really interesting is when they took away dapto treatment, the mutation was not maintained. So it was the same isolate, but it was not so it seems like there's a fitness cost associated with the mutation. Even though the same isolate is likely there, so the patient's likely colonized with this Staphylococcus argenteus. So I think a combination of all of this makes it a really intellectually stimulating case for both ID docs and clinical microbiologists.
I think I would say that this is the glimpse of the future of clinical microbiology and ID, where we now identify new species, but the genomic information help us to identify other things that we could not do it before. And we can do it quickly, potentially. That will completely revolutionize and personalize medicine and the use of antibiotics. And I think that's where we need to head on. And that's what I think the microbiologists of the future is heading on, which I think it will be a bioinformatician with a very strong clinical background.
That's exactly, yep.
Yes. But also, I wonder, and this is just a philosophical question, I guess, how complicated is going to get the ID, activity if we keep discovering new species and without looking at what's common, and if it's a treatment, why bother us, with naming a different species every single time? Are we looking, for instance, in this paper at the emergence of the Staphylococci complex, as now suggested for other species like Enterobacter cloacae complex, Acinetobacter baumannii complex, Stenotrophomonas maltophilia complex now, I don't know.
That's the beauty.
I think that's true. Yeah.
That's the beauty of infectious diseases. I always tell my residents and fellows, this is the only specialty when you have a new disease every year. No other specialty in medicine has this. So we are blessed because we are learning new things every time.
And I think it's intellectually stimulating from a perspective of when is it valuable to know the species, and when do you actually need to like get down to the granularity versus like when can you move on. But I think whole genome sequencing is, again, a big argument, not just for AST, because I know there's arguments for using whole genome sequencing to guide antimicrobial therapy, but I think this shows that there's a good argument to guide organism identification too, because if you just look at the reference genome, it's not Staphylococcus aureus. It's not nearly as similar. But a MALDI-TOF, you're restricted to what you know, which is not a lot, apparently.
And to add to that, I mean, we also still have a big gap on how to translate the genotypes of these new organisms to, clinical outcomes. So lots of work to be done in the future. And now.
Okay.
Let's move on.
Okay, I'll switch to an antifungal compound next. It's novel. This next paper is presenting the preclinical development of a novel antimicrobacterial compound designed for treatment of tuberculosis. It just entered clinical trials, called OPC 167832, so it doesn't have a fancy name yet. It's a novel carbosteryl derivative with potent antituberculous activity as a DPRE inhibitor. This paper is by a group in Japan. So tuberculosis, as we know, is one of top 10 causes of death worldwide, and it's preventable and it's treatable. But the problem with treatment is it's very intense. It takes months. So, 6 to 9 months for regular susceptible TB with at least 2 drugs. And then for drug-resistant TB, the treatment is even longer, 18 to 20 months. And these drugs that we use currently are often toxic, and patients don't tolerate them well. Just for comparison, as infectious diseases clinician, I take, an average infection I deal with would take 2 to 8 weeks to treat. So there is a need for compounds for not only drug-resistant TB, but also for drug-susceptible TB to help the eradication effort, or at least the substantial decrease of these infections in the world. So this group introduces this compound as a potential solution to that might hopefully that's active against TB and hopefully shortens therapy. It's a carbosteryl derivative. So what that means is it's likely well absorbed orally and very well distributed and metabolized by the body. Other similar drugs are fluoroquinolones, which we know we can take by mouth and get a very good levels in the blood, but also things like indomethacin, which is an NSAID, kind of ibuprofen cousin, is also carbosteryl type drug. So the mechanism of action of this new compound is through inhibition of this DPRE1 enzyme, which is basically an enzyme essential for cell wall synthesis. And the paper basically shows that this compound has bactericidal activity in vitro against and has very low MICs against TB. It has pretty low frequency of resistance development and does not antagonize other current agents used in TB treatment, so it can be used in combination. They did use a mouse model of chronic TB to show and it basically replicated similar results what we saw in vitro. That's very active. Not only that, they also identified the best partner combinations for this drug, which is delamanid. It's another new TB medication, and it also bedaquiline and levofloxacin seems to be performed the best in mouse models. Now, it's not incredibly better than our standard therapy, RIPE, but still, the evaluation was only with a few animals. So I think once it enters clinical trials, we will see better, what to expect. Can we truly shorten the therapy, or is it that much better at the eradication of the bug in patients? So I'm looking forward to see how this drug does.
This is a beautiful paper, and this is one of the examples that I get like a poster child paper, in which I see a molecule that gets discovered, and there is really nice work to try to understand the mechanism of action, mechanism of resistance. But moreover, as Alina pointed out, combination with other drugs, because we know tuberculosis is a drug, is an infection you need to treat with several drugs. So really a comprehensive paper that's the quality of papers that we want in AAC.
Yeah, absolutely. I actually was jealous of the very nice MIC data. These numbers are, like super low, astonishing. Like
I've never seen anything this low.
Right? Exactly. I was like, is this real? So, yeah. I mean, it's very promising and much needed.
The other thing I'm curious, I know the primary target for this drug is TB, but thankfully in the US, the rates are overall much, much lower than in other countries. But I would be curious to see how it works against non-tuberculous mycobacteria for our cystic fibrosis or lung transplant patients. That would be a great option, to supplement current therapies, which are few at this time.
Right, like against abscesses. That is becoming more and more important, yes.
And absolutely. Particularly the XDR tuberculosis, where we need new drugs. So there is an investment on this field, and I think that we're going to see more developments.
For sure. Okay, Ayesha, you're next.
Okay, I'm going to go back to drug discovery. But I think this is... I actually find it interesting because I think all the papers that have been picked around drug discovery are about thinking about it differently, thinking about it in a way that hasn't been thought about before, which I think is the allure of this paper, which is titled Mechanism of Action, Classification of Antibiotics by Global Transcriptome Profiling by Aubrey O'Rourke et al. At La Jolla at the J. Craig Ventler Institute in California. And so, like the previous paper we talked about, used biosynthetic engineering to exploit that as a platform for potential drug discovery. This paper looks at transcriptomics and how we can exploit the novelty there. And I know with transcriptomics, everyone's kind of used to sometimes skimming past that because everything looks like a mess when it comes to looking at whenever you treat a bacteria with an antibiotic, everything is pretty much upregulated. And I think that's part of the problem that they're trying to circumvent. So this paper specifically looks at transcriptomics as a affordable mechanism for understanding mechanism of action of antimicrobials with novel chemistry and hopefully circumventing traditional analytical chemistry techniques. And the ability of transcriptomic profiling to provide this antibiotic fingerprint has, I think, recently inspired a bunch of studies, especially with increased technology when it comes to sequencing, RNA-seq, to look at how can we avoid the rediscovery of known compounds, especially if we're trying to look for really novel agents. But the problem seems to be that even if we know the target, whenever we look at transcriptomics, there's a lot of overlaps between different classes of drugs, because usually you're looking at the downstream effects rather than the initial target. And I think people recognize these as like protective stress responses are always upregulated. So I think this noise sometimes can distract people from what the actual mechanism of action is, which is upstream. And that's been part of the problem. So what this paper does is they use E. coli and next-gen sequencing to treat E. coli with 37 antimicrobials that are binned into distinct categories of mechanism based on their mechanism of action. And these are all known mechanisms, and use that as a foundation almost for machine learning, and see if we can then feed use this as a tool to predict the mechanism of action of something else. And I think that is really interesting because it's all about saving time and resources. So they use the methods I actually do want to say are interesting. They use an E. coli strain that's deficient in efflux pumps as well as has an outer membrane that's compromised. And this was intentional so it could kind of work physiologically as both a gram-positive and negative, which I thought was interesting. And they just treat this E. coli strain for 30 minutes with these 37 antibiotics, and they're binned into categories. So overall inhibition of DNA replication, RNA synthesis, protein synthesis, cell wall synthesis, and cell membrane function, as well as fatty acid synthesis. So just these are 6 distinct categories. And I think they do come up with something valuable, which is very different than what we're used to. So the first thing is, because they treat for 30 minutes, they're trying to narrow down the window to get things that are upregulated directly related to bacterial killing as opposed to downstream effects. And ultimately, what they discover is, I think, important distinct signatures. So for example, they discovered that the upregulation of a colonic acid synthesis gene cluster is a traditional marker for antibiotics that target lipid 2 and peptidoglycan synthesis. So again, it's a secondary marker, but it's something that's very, very unique to antibiotics that target lipid 2. And ultimately, what they end up doing is narrowing down to a set of around 440 genes that are useful in predicting the function of known compounds with about 85% accuracy, which I think is pretty interesting. And the ones that they missed were antibiotics like polymyxin B, and I imagine Dapto would be mixed too. And the reason they missed it is because these antibiotics have multiple targets rather than targeting a single cellular process or pathway, which I think can be really confusing for algorithms like this. But ultimately, the paper shows that if we fine-tune transcriptomics to see what is really unique and what is just cut out the noise essentially and use machine learning, which I think people are using more and more now with next-gen sequencing, that we could potentially use this as a diagnostic tool to predict and classify novel antimicrobials and not replicate efforts when it comes to drug discovery.
Yeah, this is another paper that I love. This should be called Understanding Transcriptomics. That's for the first time really see very clever approach to use big transcriptome data in something really useful. And that could potentially even be clinically useful in antibiotic susceptibility testing. So I think that's great. That's a great idea. Very innovative.
And I thought about also it could be used, I think, from a clinical microbiology perspective. People are looking into both bacterial and host responses, like host immune responses in response to infection used in the process of diagnostics. And I think something like that, because RNA-seq is like ideal for analyzing host responses too. So I think there's a lot of potential for the same type of technology.
Yeah, that's an interesting point, kind of looking at the host response, because we know there are some bugs like Acinetobacter. It's very difficult to say, is it truly causing an infection or is there for, just there for a ride? And maybe looking at the host response can tell us what's happening.
Right. Even the accuracy of our susceptibility testing methods, are they giving us the real phenotype of the bacteria, or is going to be more severe or less in the actual infection side? Right. So I think there's a lot of potential, yes, in the transcriptomics and all the metabolomic, studies that regard. Okay, Alina?
Okay, the next one paper I'll talk about is lactoferrin as a broadly active against yeast and highly synergistic with amphotericin B. By a group from Australia, by Kenya Fernandez. This was a very long paper, and the group did an amazing, very, very thorough job at looking at lactoferrin's role in treatment of yeast. This compound has been studied for a long time, and the results have been very mixed. And the group in this paper addresses possible reason why we've had mixed results with this compound. So, they first started looking at different sources of lactoferrin. They looked at the 3 different suppliers, and they found that the lactoferrin products differ in their iron saturation as well as amount of digestion products present in those compounds, and they found that actually affects how your susceptibility testing and how yeast will respond to this compound. So why is this important? So the mechanism of action of lactoferrin is it's a big molecule, and some of it is thought to be due to sequestration of iron from the yeast, limiting the growth. But other other groups also shown that lactoferrin is broken down in other smaller peptides, including antimicrobial peptides that might have a role. So the iron, that's why the iron saturation and the presence of those breakdown products matter. So then they kind of defined these compounds, and then they looked at each of the different lactoferrin supplier compounds. They tested against different yeast, and they really focused their investigation on Candida, Cryptococcus, and Saccharomyces. They tested for synergy with amphotericin and other antifungal drugs. And they found that the synergy was really truly with amphotericin only, and not other antifungals. And this was repeated in the waxworm model of infection. And then they also confirmed that there is still variability in the treatment response due to this, due to actually the peptide compounds, but not iron sequestration. And finally, they also looked at the kind of mechanisms of actions beyond iron sequestration, because that's really kind of seems to be thrown out a lot as a mechanism, but they say that there is actually a direct fungal cell interaction between lactoferrin and the yeast. And in Candida, it appears to be preventing biofilm formation, and in Cryptococcus, it reduces capsule formation when used in conjunction with amphotericin. So in summary, despite the years of inconsistent results, lactoferrin and its derivative peptides still might be used as potential therapeutics for fungal infections. I was just very impressed with the amount of work and thoroughness of that. They really tried to control for everything in their study and kind of tried to sort out the controversies in the field.
Thank you, Alina, for that thorough. This is a very dense paper.
It was.
Interesting. This lactoferrin has been trying to be used in different situations because it's a natural product. It's in milk in developing countries. We know that that may help immunity and all that. And finally, you see a very interesting word thinking that this could work against yeast, and very thorough, even mechanistic studies that will help understand a little bit more.
Yes. So again, we are looking back at molecules that have been studied for years in aims to maybe this time we're going to tackle the mechanism of resistance or mechanism of action, and then put it back in put it in the clinic practice. I think I am more used to work with bacteria, and that's the case with colistin. We were short of new molecules, so let's go back to colistin, and with the viral compound, we are in a crisis with COVID-19. Let's go back to the molecules that have been used for years, and let's see if it works And then so this is another example. This time with yeast, and I wonder if they will actually is going to be active against Candida auris, which is picking up. Okay, Ayesha, I think you're next.
Okay. I'm going to save the best for last, which is a clinical microbiology one. And I'm going to do another one I think this is something that was interesting to me because it is drug discovery, but looks at a novel beta-lactamase inhibitor that actually truly does have, I think, quite a significant novelty in terms of how many beta-lactamases that it can inhibit, and is something that is unprecedented in terms of what we currently have. So this was done by a group at QPEX Biopharma, which is a pharmaceutical company in San Diego. And the molecule is called QPX7728. So this is a biochemical characterization of this compound, a new ultra broad spectrum beta-lactamase inhibitor of serine and metallo-beta-lactamases. And even though I would normally look at a word like ultra broad and say maybe just broad spectrum, but I do think this is actually impressive enough to have that potential. So currently, I think we've seen a huge wave of a lot of novel beta-lactamase inhibitor combinations that have come out. that do have a lot of promise, I think, especially when it comes to having reversible activity where you're not just inhibiting one compound, but you're one beta-lactamase, but you're able to do multiple classes. So this paper is trying to look at how do we address metallo-beta-lactamase producing carbapenem-resistant Enterobacter ACA, which I think is the one big challenge we don't really have a very good answer to. Metallo producers are resistant to the novel beta-lactamase inhibitors that have been recently introduced. So that includes avibactam, which is paired with ceftazidime, vaborbactam, which is paired with meropenem, and relbactam, which is paired with imipenem. Though they've been, I would say, instrumental in combating KPC producing CRE. These novel beta-lactamase inhibitors are particularly important, I think, because they do introduce a whole new class of beta-lactamase inhibitors that have been effective in having reversible activity, so recycling, and they're able to therefore inhibit multiple classes of beta-lactamase within the same cell. And still, there's a lot of inhibition, I think. There's a lot of limitations. So beyond metallo-beta-lactamases, they're ineffective against class D enzymes in Acinetobacter baumannii. So there's still a lot of holes to be filled. So this paper looks at this compound, which they call as having ultra-broad spectrum activity because it can inhibit a broader series of serine and metallo-beta-lactamases. From the same classes, and it's made from the same classes of boronates that were the basis of vaborbactam, so structurally similar. So they do show, and all of this is using purified enzymes in vitro, that this compound can inhibit class A ESBLs with very low IC50s, comparable to carbapenemases like KPC, and having equal, I would say, having equal potency or higher potency than avibactam, relbactam, or vaborbactam, which was interesting. Additionally, it also inhibits class D oxy-48s, which are common in CRE and also co-occurring with a lot of other class A as well as class B metallos, and OX enzymes in Acinetobacter, in addition to metallo-beta-lactamases like NDM, IMP, and VIM. So the novelty of, like I said, of these new classes of beta-lactamase inhibitors that have come out is that they have covalent binding, but do so in a reversible manner, which allows the enzyme to recycle itself. And the same goes for this enzyme because it is the same biochemical backbone as vaborbactam. The only exception being KPCs. KPC beta-lactamases can slowly inactivate avibactam over time, though the effect that this has on its activities is debatable. So overall, I think this shows that there is real potential in this new beta-lactamase inhibitor that can be combined with beta-lactams from ceftazidime to meropenem. It does definitely have superior activity to all new existing beta-lactamase inhibitors that have just come out, and those that are actually in the final stages of the pipeline as well, like durlobactam and teniborbactam. And it's specifically the novelty here is the class Ds from Acinetobacter and oxy-48s, as well as our metallos, which I think is something that we're still struggling with. So that's what this paper is.
Very hot topic, Dr. Mojica. This is your area of expertise. What do you think about it? Yeah. Beautiful, super ultra.
Ultra broad spectrum.
Yeah.
I like that. Yeah.
So what do you think, Maria Fernanda?
I am super excited about this molecule, QPX. I think, just looking at this chemical structure is like the bare minimum common structure able to inhibit all beta-lactamases, so that, and actually the authors say that that's actually the key, that is so small, it's just comparable to the two rings of the beta-lactam ring. That is the minimal possible, chemical.
It's found the conserved. It's like the conserved part of every
minimal.
The minimal inhibitory unit. Let's put it that way.
Yes, exactly. And so yes, I was very excited when I actually talked to the senior author of this paper when they were presenting QPX and ID Week, I think it was 2 years ago.
And they did. Yeah, I remember this at ID Week 2 years ago. So
yes.
Some of them work out.
Exactly. And I was very impressed with all the work that they did. In this paper is just presented the biochemical characterization, while other papers have looked at the microbiological activity and so on. And yet it cannot inhibit some of the other metallos that are present
Correct.
in clinically relevant microorganisms, such as the L1 of the Stenotrophomonas multivilia. And now the key question is why?
Why not? Because it has the same backbone. Yeah.
Well, that's a question for another podcast.
Exactly.
Let's move on with Alina with the next paper.
Well, since we're on the topic of ultra broad spectrum inhibitors, I'll talk about the safety and pharmacokinetic characterization of Nacubactam, a novel beta-lactamase inhibitor, alone and in combination with meropenem in healthy volunteers. So I'm assuming this is a novel beta-lactamase inhibitor that's a bit further in development than the one that we talked about now. It's actually kind of this novel class that we can also call enhancer, because it doesn't only inhibit the serine beta-lactamases, the class A, C, and some class D enzyme, but it also inhibits penicillin-binding protein 2 in Enterobacteriaceae. So it's truly like a true carbapenem-resistant Enterobacterialis drug that can exert direct antibacterial effect, as well as enhance activity of a co-administered beta-lactam agent. So currently it's being developed together with meropenem. And basically when this molecule is combined with the other beta-lactams, its activity expanded to also cover metal beta-lactamases and some oxa carbapenemases as well, class D enzymes. So the study in itself is a single ascending dose and multiple ascending dose placebo-controlled study to basically assess safety and tolerability of the drug alone and in combination with meropenem. It is basic, and also pharmacokinetics, and it basically shows that there's really only minor adverse events, a lot of them associated just with IV placement, like IV, pain at the site, infiltration of the IV, and then some headaches. There were no serious adverse events. There was a predictable relationship between dose escalation and drug levels. It's cleared by kidneys, so it's very similar. It behaves very similar to other beta-lactamase inhibitors that are out on the market. They also showed that there is no effect on the QT prolongation, which is escalation we always worry about in antibiotics. So, and then pharmacokinetics worked very, very nicely with meropenem. So, the levels were correlating. That means that you're not gonna, at the end of your, 4-6 hour period after administration, you're not gonna run out of your one of your drugs in the body. So they would work together the entire time. Very, it's going on to clinical trials, and I'm excited, to use it. And to why, we all seen that it seems like resistance begets resistance, and we often see that there are multiple multiple enzymes that can coexist in the bacteria, and oftentimes we don't know just looking at the antimicrobial susceptibility testing, and not all centers have the access to tell you is it NDM enzyme, or is there NDM and ESBL, or is there AXA and NDM? So it's nice to have this kind of agent where you don't have to necessarily if you don't have a rapid way to identify mechanism of resistance, and you don't know what the epidemiology of your bug is.
Another possibility in our armamentarium for the future. Any comments?
Right. I think that now the biggest challenge is going to be on antimicrobial stewardship, to decide which molecule to use when. And
absolutely, and hopefully, but we will have drugs. That's a way far cry from what we were about 5 years ago.
Exactly.
And I'm so excited about that, and how to place and deploy these drugs is going to be crucial. Right. So let's go to the last paper for Ayesha.
All right. So this paper is I think the one I was most excited about because it's addressing the age-old argument between genotype versus phenotype. So it's titled Reconciling the Potentially Irreconcilable, which I think is cool. Genotype and phenotype, genotypic and phenotypic amoxicillin clavulanate resistance in E. coli. And this illustrates, I think, an example of why we need both personalized medicine to guide antimicrobial therapy in terms of using whole genome sequencing technology in the clinical microbiology lab, but also shows the importance of understanding the basic science behind mechanisms of resistance and not just relying on traditional phenotype or even just presence or absence of an AMR gene. But the granularity is really important. I know that's pretty much the basis of my PhD. So I think this was interesting to see some of the big problems that are created if people don't look at the details. So it uses one example, which is amox-clav resistance in E. coli, which we know is a major problem, especially when it comes to drug-resistant bloodstream infections. And it's a problem mostly because it's the most commonly used antibiotic. I don't know worldwide, but I know in Europe for sure. And consequently, there's significant uncertainty in understanding how to really call something as amox-clav susceptible or resistant. Largely because there's so much variation between agencies like CLSI and UCAST. So labs get confused and they're switching over from CLSI to UCAST, and that alone led to increasing numbers of amox-clav resistance just based on a lot of labs switching over from different breakpoints and guidelines. So I think this also shows some of the existing problems which we still have to address, which is standardizing and coordinating clinical microbiology standards between here in America as well as the rest of the world. So they approach this in an interesting way, which is they try to look at using whole genome sequencing, which identifies genetic determinants, but also trying to see if you can look beyond just presence and absence of genes. So what they do is they look at both whole genome sequencing derived genotype as well as amox-clav susceptibility phenotype in a large set of E. coli bloodstream isolates collected in Oxfordshire. So this is in England from 2013, 2015. And their whole goal is to see if they can get a more reliable assessment than traditional susceptibility testing methods from using these set of isolates. About 1,000 strains, I think 976. And they're comparing it against phenotypes predicted by BD Phoenix, which is a commercial antimicrobial susceptibility system that is commonly used here in the US, most frequently as well by clinical labs. And I think what was really interesting is how they tried to basically see if they can predict resistance using just whole genome sequencing alone and circumvent traditional AST. So first of all, at the starting baseline, about 35% of their isolates were resistant, called as resistance. And if they just looked at presence or absence of a beta-lactamase, that was not enough. The sensitivity of that was 23%, which is extremely low. But when they started to understand mechanisms of resistance a little bit more, so they included genetic features associated with penicillinase hyperproduction, for example, so mutations in promoter sequences or copy number variation, then the sensitivity went up to 82%, which I think is very telling to how much the value of understanding those details are. And interestingly, almost all of the discrepancies, the things that they didn't get right, existed pretty much between MICs that were within one doubling dilution of the breakpoint. But that's expected in antimicrobial susceptibility testing. That's where we have the toughest time telling apart what's right and what's not. But I think the most important thing that they demonstrated is once they were able to redo the reference, so they basically did agar dilution again and didn't rely on the BD Phoenix, they actually got 97% prediction rate, which is extremely high. And all of the MICs were within essential agreement, which means that the raw value predicted was within plus or minus one doubling dilution. And I think that shows how much value there is in trying to circumvent some of these inherent genotypic, phenotypic problems that we have, which largely come from lack of understanding, I think, mechanisms of resistance. I think the paradigm often is that beta-lactam resistance is generally due to the presence of a specific beta-lactamase. But in this case, they're showing that mechanisms of resistance largely are multifactorial. So things like combinations of multi-copy beta-lactamase genes are important. Mutations associated with resistance in promoter regions are important, as well as beta-lactamase inhibitor resistance mechanisms. And it all adds up to give the eventual outcome. And I think that's the most novelty here. And the caveat being, when you have discrepancy between genotype and phenotype predictions, it's inevitable when you're relying on the susceptible resistant binary. So I think they also show that there's a problem with how we classify these things if we're trying to reduce everything to one cutoff. I think the boldest claim that they make as a result of the study is, what if we try to avoid breakpoints entirely, which I know is a really bold claim, because that is inherently a big problem. And alternatively, try to develop a machine learning algorithm that can use whole genome sequencing to predict MICs entirely. And at least they showed, using the set of 1,000 isolates or so, that it is potentially possible with a 97% accuracy, which I think is pretty good.
Yeah, I know about this work because I'm familiar with the authors, and I love that bold statement, and I think that's how it should be. However, the beta-lactamase inhibitor resistant prediction by genomics is complex, and this is the field where we'll need a lot of work. So I think we need to move for the last, because we're running out of time. Alina, you are going to close this.
Well, now that we geeked out about all the new beta-lactamase inhibitors and repurposing of old drugs, this is a complete turn to saying maybe we should use less antimicrobials and more antibodies. It's antibacterial monoclonal antibodies do not disrupt the intestinal microbiome or its function, by AstraZeneca group. And they basically talk about how the problem with antibiotics is that they, even though they work amazingly to treat this complex infections, that they save lives, they also do some damage. Specifically, they drive emergence of resistance, and that's why we have to talk about all the new drugs. But they also can predispose someone to infection with other bacteria like C. diff by wiping out their the good microorganisms that we all have on our skin and our bodies. And not only it's issue from the infectious diseases standpoint, there's some suggestion that, once the microbiome metabolics are disrupted by antibiotics, you can also get predisposed to things like cancer, because the bacteria break down bile salts and other byproducts of what we eat. So this paper is basically a proof of concept that antibacterial monoclonal antibodies do not disrupt human and do not disrupt microbiome. The study is actually in mice, so not human just yet. So the group looked at the monoclonal antibodies against common pathogens like staph, Pseudomonas, and Klebpneumo, and they compared them to the effects of vancomycin, meropenem, linezolid, and fluoroquinolones in the mouse model, and not unexpectedly, antibodies did not disrupt the diversity of mouse gut microbiome, and also did not alter the metabolic functions of the microbiome. So that said, it's good results. I'm not surprised by them, but I think it was important to show. I'm still would like, I don't think antibodies are that close to be used in clinic just yet, because we still have to see how good they are against the actual infections. So we'll see.
If we've learned something in COVID-19 is that the antibodies may work in certain stages of the disease. If that applies to bacterial diseases is a matter of debate. But I think it's interesting that people are looking at that very carefully.
I think it makes a general argument for narrow spectrum, right? Narrower and narrower.
Yeah. Dr. Mojica, why don't you close this session?
Well, okay. So I would like to thank you, Dr. Ayesha Khan and Dr. Alina, I cannot pronounce your name. I'm so sorry. You know, like I said in one conference, I'm Colombian. I cannot pronounce certain words. And thank you, Dr. Arias, for this nice invitation. I think we all had a great time, and we discussed very interesting papers.
Thank you very much, everybody, for your input and your work on this. And this is Cesar Arias, Editor-in-Chief of AAC, signing off. Thank you very much.